The Ünal lab is sort of broadly interested in generation of gametes through the process of meiosis. And in order to produce gametes with the highest viability, cells need intricate quality control pathways to prevent inheritance of damaged or defective materials. And that includes organelles. So today, Professor Ünal will tell us about her lab's recent work determining how cells ensure that gametes inherit functional nuclei during meiosis.
Thank you so much for the introduction, Ross. And I want to thank all the organizers for putting together this fantastic meeting and also inviting me to be a part of it. So I consider Carnegie as my scientific home. This is where I did my graduate research in Doug Ocean's lab. And this is the place where I learned how to do fundamental research and creative research. So it's really good to be back home, even though it's on a virtual platform, and see so many familiar names in the participant list. So what I want to talk to you today, or tell you a story, a fully unpublished and new story that my lab has been working on, and that revealed an interesting mode of regulation to nuclear pore complexes that Patrick has beautifully introduced us today. And we gained this information or biological insight through studying a truly fascinating developmental program. And of course, the program I'm referring to is meiosis, or meiotic differentiation. This is the process by which sexually reproducing organisms, such as this cute pair of cats, give rise to gametes in the form of egg and sperm. And so to study this process in molecular detail, we use a simple and genetically tractable model organism, budding yeast Saccharomyces cerevisiae. Just like you and I, yeast cells also undergo meiosis to produce gametes. So here's a roadmap of the stages that a cell undergoes as it proceeds through the meiotic program. A diploid progenitor cell first duplicates its genome during S phase. This is followed by a lengthy prophase 1 stage where several changes occur to the chromosomes in the form of programmed double-strand breaks, homolog pairing, as well as crossover, which then culminates in their segregation through 2 consecutive nuclear divisions called meiosis 1 and meiosis 2, and a final stage of gamete maturation, which then eventually leads to 4 haploid gametes, which in yeast are also referred to as spores. So I want to make a few remarks about this program. So all these sort of different changes that we're seeing that occur during meiosis are undertaken and regulated by a very dynamic and tightly controlled gene expression program. And in budding yeast, we can study this at very fine time scales, because we have the ability to induce meiosis at will whenever we want, and then we are able to study it in a synchronous manner in populations of cells, as well as we're able to do live cell imaging through the entire course of meiosis, which gives us information about both single cell and population-based analysis. Second, in addition to the changes that we see that occurs to the chromosomes, there is also extensive organelle remodeling and quality control that takes place during meiosis. And in essence, the way I think about this is a precursor cell actually undergoes a programmed self-destruction event as it creates 4 new cells from the material that it degrades. So it's a very sort of parsimonious recycling and recreation of life that takes place in this developmental program. So there's a lot to learn besides what happens to the chromosomes. And finally, meiosis has this amazing feature built into it, in that it has this ability to rejuvenate the cells. What I mean by that is, independent of the age of the progenitor cell, the gametes that are formed at the end are born young, independent and devoid of any of the age-induced damage that was present in the precursor cell. So my lab is interested in understanding and studying all these different aspects of meiosis in budding yeast. But today, what I want to talk to you about is regarding one particular remodeling event that we uncovered by studying what happens to the nuclear pore complexes during the course of meiosis. And that's what I'm going to be focusing on in the remainder of my talk. So Patrick already gave a beautiful introduction to the nuclear pore complexes, and I'm just going to briefly go through this slide. So these are amazing macromolecular machines that regulate nuclear cytoplasmic transport. It's conserved from yeast to humans. It has this eightfold symmetrical cylindrical structure, and roughly divided into 6 subcomplexes. And these are basically listed here, ranging from cytoplasmic knobs, channel knobs, Y complex, transmembrane knobs, nuclear basket, and the inner ring. And one thing to note is several members of the nuclear pore complexes, especially those that are integral parts, such as the transmembrane knobs, inner ring knobs, and Y complex, which I will refer as the MPC core. These are exceptionally long-lived proteins in the cells, which means that in a post-mitotic state, they stay with the cell from the time a cell is, generated to the time that the cell dies. And in terms of besides the functions of regulating nuclear cytoplasmic transport, MPC is also involved in chromatin organization, transcriptional control, cell cycle progression, and RNA biogenesis. So it has this wide range of functions besides being gatekeepers within the nuclear envelope. So we became interested in looking at nuclear pore complexes, again, from our interest in understanding meiotic differentiation and how different organelles and compartments within the cell behave during this process, as it has this natural rejuvenation capacity, right? And also given the fact that several members of the nucleoporins are long-lived proteins, and as Patrick mentioned, for budding yeast in mitotic cells, as well as in meiotic cells, the nuclear envelope doesn't break down. So when a nuclear pore complex is created and inserted within the nuclear envelope, it stays there, as the cells undergo cellular division. So we wanted to see first what happens to the nuclear pore complexes. And this project, this only published data that I'm going to share with you today is a story that actually initiated by 2 fantastic graduate students in the lab, Grant King and Jay Goodman. And what they were interested in looking into is to understand, again, how nuclear pore complexes are regulated in the cell, and whether or not this sort of changes that occur to the nuclear pore complexes may have anything to do with the meiotic rejuvenation phenotypes that we observe during the course of meiosis in budding yeast. So what I'm showing you here is a montage, and we're going to see a couple of these throughout the talk. But what I want to walk you through is what's going on in here. We have chromatin marked in magenta by a histone. So these are live imaging samples of montages, and in green is a subunit of nuclear pore complex, in this case, NAP 170. This is part of the core nuclear pore complex member. And what we see is that in pre-division, it's not that exciting in the sense that this nucleoporin decorates in this perinuclear region. During meiosis 1, when the nuclei divide, the nucleoporin also follows that. But what happens during meiosis is, as the chromosomes are segregated, the nuclear pore complexes, and in particular this nucleoporin NAP 170, is now sequestered in this region of the nuclei that is away from this dividing chromosomes. And in late meiosis, the sequestered material is degraded as new nucleoporins are generated. So I'm going to play the movie a little bit quickly here, and you can see what's going on. And I'm going to summarize basically what we have found from this analysis in one simple slide, in that what Grant and Jay has found, that the nucleoporins and the nuclear pore complexes undergo the sequestration during the 2nd division of meiosis as the chromosomes segregate. And this occurs in a continual membrane structure. And this sequestered material gets eliminated, and we name this material GUNC, for gametogenesis uninherited nuclear compartment. But I also want to indicate that this event occurs in both young and age cells undergoing meiosis. And in age cells, what we see is that many of the nuclear damage in the form of nucleolar aberrations and protein aggregates are also sequestered into this GUNC structure. And so this is a way by which the gametes exclude these age-induced damage. So we're actively looking into how this process is regulated. But by studying this nuclear pore complex remodeling in the context of young cells that are undergoing meiosis, we also found some very interesting biology, and this is what I want to share with you today. So what Grant did, based on his interest in looking in the nuclear pore complexes in a more extensive manner, is he endogenously tagged several older nucleoporins with a GFP marker within these 6 subcompartments and started looking in how do they behave. And what we found is similar to what I've shown with regards to this inner ring NUP 170. Many of the subcompartments, all of the other subcompartments, except for the nuclear basket, behaved like NUP 170. But the nuclear basket nucleoporins behaved very differently. And that's shown in here. What Grant has observed is when he actually tags a nuclear basket versus a nuclear pore complex core component. Here, we're looking at these FG NUPs. What he sees is that the core of the nuclear pore complexes get sequestered, whereas the nuclear basket actually segregates together with the dividing nuclei. So this suggests to us that the nuclear pore complex actually maintains some form of modularity. And we wanted to understand how this is regulated and what does it mean biologically. So to get a little bit more understanding into this sort of form of modularity, Grant also undertook some experiments by using super resolution microscopy. And this was done in collaboration with Sue Jasperson's lab in Stowers Institute. And what Grant found in this case is very similar to what he found in his live imaging data, in that the basket nucleoporin versus the core nucleoporins behave very differently. Whereas the MPC core is sequestered during meiosis 2, as shown here, the nuclear basket nucleoporins are actually segregated together with the gamete nuclei. So a little bit more introduction about what is the nuclear basket. So this is a part of the nuclear pore complex that is composed of 5 different types of nucleoporins, NUP1, 2, 60, MLP1, and 2. And I think about it as almost like a bridge between the chromatin and the core of the nuclear pore complex. And it associates with both the chromatin and the MPC core, and the way by which the basket binds to the MPC core is through associations with the Y complex. So the first question that Grant wanted to ask is, how is MPC modularity regulated during meiosis? And to get at this question, what he first decided to do is to perform some live cell imaging experiments with higher time resolution, looking at every 5 minutes throughout the course of meiosis to see how the nuclear basket subunits behave. And with that, what he started observing is something very interesting that happens with NUP60 and NUP2. And I'm going to be showing mostly NUP60, but NUP2 pretty much behaves the same. And I want to draw your attention to this sort of yellow bracketed area here. And this is, again, a montage scanning through meiosis, and 0 marks the time of the second meiotic division, where we see the sequestration event for the core. And this happens, again, very transiently for the basket, but quickly afterwards, the basket actually returns back to the gamete nuclei. But what Grant has observed is, long before this event actually starts, during the time of metaphase 1, what he actually sees is that this transient dissociation of the nuclear basket subunit NUP60 from the nuclear periphery into the nucleoplasm. And this may be even more apparent when we look at single Z stacks. As you can see, it goes from this peripheral to a nucleoplasmic localization. So this suggests to us that actually the modifications or the regulations that start happening that establish this modularity actually occur way before the second meiotic division. So we want to understand how this happens, and to sort of get at that question, what we sort of look through is a number of candidate regulators that we know to be active during the stage of meiosis metaphase 1. And one attractive candidate was the polar kinase CDC5. This is a conserved kinase. In budding yeast, it's called CDC5. It has many important functions in meiosis that are important for regulating chromosome dynamics, holiday junction resolution, cohesion dissociation from chromosome arms. So it was at the right time that this kinase was present. And we also know from work of nuclear pores in other systems that nuclear porins undergo extensive phosphorylation. So we wanted to see whether or not this kinase might do something to NOP60 to regulate its modularity. So to get at that, Grant did this experiment where he did some live imaging experiments again, initiating meiosis and starting the live imaging around 4 hours, and run the cells into a metaphase 1 arrest by blocking an anaphase promoting complex regulator CDC20. So this way we can actually accumulate all the cells during metaphase 1, and then he can track then the sort of by looking at the position of the spindle, which is the centrosome equivalent organelle, we can actually see sort of stage these cells relative to where they are in meiosis. And a separated spindle pole to us would basically be indicating that the cells have actually arrived at metaphase 1. So what we expect to observe is in a CDC20 arrest, at the beginning of the live imaging, we expect to see this perinuclear localization. But as cells get arrested during CDC20 arrest, and then we expect to see in wild type this nucleoplasmic localization going from a peripheral to a nucleoplasmic phase. So the question is what happens if we take away CDC5? So both CDC20 and CDC5 are essential genes, so we can't just delete them. So we generated meiosis-specific depletion alleles for them to do this experiment. So when Grant did this, the results were quite striking, and I'm going to share some of his movies with you. In the CDC20, watch until the spindle poles separate. What we can see is that you get this nucleoplasmic localization as the spindle poles separate. But in the absence of CDC5 function in meiosis, following spindle pole body separation, as cells arrive biochemically to metaphase, what we see is that there is still maintenance of NOP60 at the nuclear periphery. So this told us that CDC5 is necessary for dissociating NOP60 from the nuclear periphery. And the second question we wanted to ask to follow up was whether or not we can observe any concomitant changes in the phosphorylation status of NOP60. And this was the case. In CDC20 arrested cells, we can see during the course of metaphase arrest, we can see a phosphoshift that is diminished when we take away CDC5 from these cells. And the follow-up obvious question that we wanted to ask and Grant asked is whether or not CDC5 polokinase was sufficient to promote the dissociation of NOP60 from the nuclear periphery. So to do this experiment, Grant used a genetic background where we can permanently arrest cells in meiotic prophase. So in this case, normally wild type cells would not have any CDC5 kinase activity. But what we can do then is to use an inducible promoter to, basically induce CDC5 whenever we want, and induce either a version that is kinase dead or a wild type version. So what we're doing is basically the only thing that we're changing in the state of prophase arrest is whether or not the cell has a functional versus a kinase dead version of polo. And so the expectation is that if we basically don't do anything in wild type, we expect to observe this peripheral localization for NOP60. And if CDC5 kinase activity is sufficient, then what we expect to see is this switch from a nuclear peripheral to nucleoplasmic localization. And here are some more movies for that. So with the kinase dead, the control experiment first, what we see is not very exciting in the sense that NOP60 remains very peripheral throughout the course of the experiment. But what Grant has found is upon induction of CDC5, which the movie is looping and it's right around that time, I hope you can appreciate that NOP60 becomes nucleoplasmic very soon after CDC5 induction, suggesting that this kinase is both necessary and sufficient to promote NOP60 dissociation from the nuclear periphery. And again, concomitantly, what Grant has observed is phosphoshifts in NOP60 that are dependent on expression of induction of CDC5, again, suggesting the involvement of this kinase in regulating this nuclear basket component. So the question then that becomes is what are the critical NOP60 phosphorylation sites? And in that, we established a collaboration with Joao Matos's lab in University of Vienna, along with his graduate student and now postdoc Rahel. And what they have done is a series of mass spec experiments to actually look through phosphorylation status of many of the nucleoporins during the course of meiosis. And these are quantitative mass spec experiments. And one of the things that they have done is to do a very similar experiment of this prophase arrest in meiosis and really inducing either wild type or a kinase Z. They have also done extensive time courses. So we have very spatial time resolution in synchronous meiotic cultures. So the culmination of these experiments, what they have found is a total of 9 different phosphorylation sites in NUP60. And I want to map them onto the cryo-EM structure to demonstrate where they actually are located, because this reveals some very interesting information for us. So we're looking at the cryo-EM structure, and I want to zoom now on one of the single spokes. And in the dark blue, we're looking at the Y complex. Again, this is part of the nuclear pore complex core. And light blue marks the nuclear basket, and the phosphosites for NUP60 we're marking in magenta. And if we zoom in, what was so interesting to us is that the NUP60 phosphorylation sites occur right at the interface where the nuclear basket faces the Y complex. So this was quite interesting because it suggested that this phosphorylation may be modulating the sort of dynamics and localization of NUP60. Naturally, Grant then generated a number of different phosphomutants. So the phosphorylations occur at both the N and C termini of NUP60, and he created a combination of mutants. And I want to share data from one of such, this 9A mutant that lacks 9 phosphorylation sites in NUP60. So again, in the wild type cells, the experiment is similar to what I've shown before. We're arresting cells in prophase, inducing polokinase. And what we now see in wild type, right, when we induce polokinase, that NUP60 goes from peripheral to nucleoplasmic. However, when we look at this 9A mutant, even though we induce, CDC5, very similar to in this condition, in the 9A mutant, now the NUP60 maintains its more peripheral localization, suggesting that these phosphorylation sites are important to regulate, again, this dynamicity of this nuclear basket subunit. So to summarize what I've shown up to here, which will be the bulk of the talk, is that the nuclear basket versus the core have very distinct behaviors during meiosis, and that the CDC5 polokinase promotes the dissociation of NUP60 and NUP2 from the nuclear periphery, and that these phosphorylation sites map to the interface where the nuclear basket NUP60 subunit faces the Y complex. So the question you may ask is, okay, this is great and all, but what is the biological role of this dissociating NUP60? Why do cells go through this hassle? And this part of the talk is going to be a little bit more speculative, but we have, I think, some exciting preliminary data that I want to share with you regarding this part. So what we observed is that, first of all, if you delete NUP60, the cells have viability problems. So the gamete viability is reduced by about 25%. And concomitantly, what Joao's and Rahel have observed is that there's increase in crossover frequencies. Okay, so in comparison to wild type cells, in NUP60 deletes, there's higher frequency of crossovers. So what does that mean? Obviously, crossovers is a natural part of meiosis. This is the way by which the homologous chromosomes are linked to one another and undergo exchange. And what I have already shared with you is CDC5 is a master regulator during this course of meiotic prophase, where it mediates crossovers, right, through multiple mechanisms. The other thing that I haven't shared with you is that during the stage of meiotic prophase, another extensive post-translational modification that occurs to the cells is simulation, okay? Several of the machinery that regulates double-strand break formation, homologous rib combination, synaptonemal complex formation undergoes extensive simulation during meiosis. And the way by which simulation is regulated is, of course, through conjugating this ubiquitin-like modifier sumo onto substrates. But interestingly so, you also need to remove the simulation. And one of the ways by which you can do so is by this protease called ALP1. And ALP1 is interesting because it has been previously shown to be associated at the nuclear periphery via its binding to NUP60 and NUP2. So what we're postulating here is that the sumo balance in the cell is important part of meiotic prophase. However, our current working model is such that upon this regulation mediated and imposed by CDC5 polo, this actually imposes the dissociation of OP1 because of its sort of obligate association with NOP60 and NOP2. As a result of this, we expect to see changes in crossover regulation, potential other functions that are impacted by this, changes in the localization of this sumo protease. And so we're actively investigating how these changes are impacted within meiotic cells and how the CDC5 dependent phosphorylation events that occur on the nuclear basket impacts these and impacts this meiosis-specific simulation events. So with that, I would like to stop and thank many people. I already mentioned Grant, who did bulk of the work that I shared with you today, and he has been helped by a fantastic technician in the lab, Kirtana Chatlapalli. I also want to thank all members of my lab who I have the privilege to work with, as well as my collaborator and colleague Gloria Brar, who I run a team lab with, as well as members of her lab. Together, we formed the Brun Lab for Brar and Ünal. I want to specifically thank our collaborators, Joao Matos and Rahel Wettstein and University of Vienna, as well as our collaborators in Stowers Institute who helped with all the super resolution imaging experiments, Sue Jasperson, Joseph Warburg, as well as the Stowers Imaging Core, as well as the funding sources. And I want to thank you for your attention and happy to take any questions. Thank you.